Hi, my name is Abigail Kim, a graduate of Seoul International School.
And my name is David Lee, a senior at Yongsan International School of Seoul.
The name of our research is Transfer and Suppression of Antibiotic Resistance Genes in the Water Cycle.
The Han River is a major water source in South Korea, like Lake Geneva and the Yangtze River. We started this study to investigate and find a solution to the issue of the contamination of this water source.
The spread of antibiotic-resistant bacteria is a serious health threat, as these bacteria render existing antibiotics as less effective. In fact, according to O'Neill's report, 700,000 people die annually due to antibiotic resistance. They have been found in Paltang Lake as well, which is upstream of the Han River and a drinking water source in South Korea. We wondered how the water source became contaminated.
After looking at a case study where antibiotic resistance genes have been found being transferred through the air in the snow, we asked the question, could the evaporation of water also be a mechanism for these genes to be transferred over distances? Then, we also conducted a study that investigated whether there was an eco-friendly method of suppressing antibiotic resistance gene transfer.
There are two objectives of the study. 1. To identify how the antibiotic-resistant genes move around the water cycle, and 2. To research eco-friendly methods to decompose the ARGs. This experiment was conducted to determine whether the ARGs were transferred to nearby freshwater through the water cycle. The ARGs were added to the red Petri dish A every 24 hours for 1 week. The movement of ARGs was confirmed using electrophoresis. This time, the same experiment was conducted with soil, and the ARGs were also found to be moved to nearby soil. In addition, the transfer of ARGs per day was examined, and the results showed that the ARGs were found after 3 days on average. Next, the PCR test was performed to amplify the genes of water droplets on the container, and the results showed that 33 out of 36 water droplets contained ARGs. Moreover, ARGs were not detected in the evaporated freshwater after removing electrostatic attraction, suggesting that the ARGs were transferred due to attraction between water and the genes.
Next, the ARGs were added to the red Petri dish, and after 7 days, it was found that the ARGs moved to nearby soil and changed the normal E. coli present in the soil into antibiotic-resistant E. coli. Additionally, 16 cabbage seeds were planted in dishes containing the soil with ampicillin-resistant E. coli, and the leaves of plants grown in that soil were found to be contaminated by the ARGs that had been transferred. To investigate whether humic substances found in urban residential soils could suppress the transfer of ARGs caused by water evaporation, the ARGs were inoculated into the urban residential soil extract. The results showed that gene transfer to the other Petri dishes through evaporation was suppressed in the case of freshwater containing humic substances. There was further confirmation that these humic substances were able to suppress the movement of ARGs through the water cycle when PCR did not detect ARGs in the condensed water droplets collected from the container in the freshwater condition treated with humic substances.
In conclusion, we confirmed that antibiotic resistance could indeed migrate through the water cycle and could be passed on to bacteria in distant water and soil.
We hope to solve global water contamination problems caused by antibiotic-resistant bacteria using humic substances, a potential eco-friendly solution.
Thank you for watching our video.
And we hope our findings can help make our drinking water sources clean.